Okay, this is the Professor, CBMD PhD, and today is Monday, November 13th, 1923. It is 1:42 and 28 seconds, AM Eastern Standard Time, and today's research, glutathione and cancer. So let's hop to it here. How the antioxidant glutathione keeps mitochondria healthy. Date November 8th, 1923. Source, Rockefeller University. As scientists have discovered how mitochondria sense and control their glutathione levels, an antioxidant produced throughout the day, the first nutrient sensing mechanism identified for an organelle, the finding has great translational potential. The handful of these nutrient sensing mechanisms thus far identified have had a profound impact on human health. One prime example is the discovery of the nutrient sensing mechanism for cholesterol, which led to the development of life-saving statin drugs and the Nobel Prize. As these discoveries have focused on how an entire cell detects nutrients, but within every human cell are self-contained, membrane-bound organelles, all of which are equally in need of fuel to carry out important functions. Might they then have nutrient sensors of their own? The Research Laboratory of Metabolic Regulation and Genetics have discovered the first such sensor for an organelle, specifically mitochondria, the cell's power center. The sensor is part of a protein that does triple duty. It senses, regulates, and delivers the antioxidant glutathione into the mitochondrial interior, where it plays critical roles in tamping down oxidizing reactions and maintaining appropriate iron levels. They believe this is going to be a very fruitful find. Every time people have studied nutrient sensing, they have learned a lot about biology, and many drugs have been developed as a result. As glutathione is an antioxidant produced throughout the body that plays many important roles, including neutralizing unstable oxygen molecules called free radicals, which cause damage to DNA and cells if left unchecked. It also helps repair cellular damage and regulate cell proliferation, and its loss is associated with aging, neurodegeneration, and cancer. As a result, glutathione supplements have become increasingly popular as an over-the-counter approach to wellness. The antioxidant is especially abundant in mitochondria, which cannot function without it. As a respiratory organelle, mitochondria produces energy, but mitochondria can also be the source of a lot of oxidative stress, which has been implicated in cancer, diabetes, metabolic disorders, and heart and lung diseases, among others. If glutathione levels aren't precisely maintained in mitochondria, all systems fail. None of us can survive without it. How glutathione actually enters mitochondria was unknown until 2021, when the researchers discovered that a transporter protein called SLC25A39 delivers the package. It also appeared to regulate the amount of glutathione. When the antioxidants are low, the level of SLC25A39 increases, and when the antioxidant levels are high, the transport level goes down. The findings strongly suggested that the mitochondria had some sort of way to detect and adjust these fluctuating levels. Somehow mitochondria figures out how much antioxidant it has, and depending on that amount, it regulates the amount of antioxidant it lets inside. And to ferret how the mitochondria does it, the researchers used a combination of biomedical studies, computational methods, and genetic screens to discover that SLC25A39 is both a sensor and transporter at the same time. It has two completely independent domains. One domain senses the glutathione and the other transports it. The protein's unique structure may explain its abilities. When they snipped it from the protein, its transporter abilities remained intact, but it lost the ability to sense glutathione. Finding that interesting loop later led to the understanding of the sensing mechanism. The study also bolsters the theory that glutathione is a chaperone for iron, which is required for virtually all functions within a cell. Iron is not only the most abundant metal on Earth, it is also the most abundant metal in our cells. As iron is also highly oxidative, without glutathione to keep it in line, it initiates oxidative stress in cells, causing damage. They believe maintaining The glutathione to iron ratio is very important because if you have too little glutathione, then iron becomes very reactive, and if you have too much glutathione, the iron will not be usable. Their experiments determined that SLC-25A39 carries a unique iron signature on its surface as part of the glutathione sensing mechanism. Now that the researchers know how SLC-25A39's package delivery system operates, they can experiment with manipulating it. This particular transporter protein is unregulated in a group of cancers, and people have tried to change overall glutathione levels, but now they have a way to change it in mitochondria without impacting other parts of the cell. And this kind of targeted therapy could potentially lower the number of side effects that can come with altering glutathione levels across the whole body. I can see a lot of translational outcomes leveraging this new understanding. And this is a professor of microbiology, bioinformatics, pathophysiology, and leader of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.